suppressor genes. Now, these type of genes are responsible preventing the cancer. They prevent the formation of the tumor. Normally, there are 50 type of human cancers caused due to the mutations occur in this type of gene. P53 is a human genome guardian, also called the Death Star, because it prevents or it fights against the mutations that occur in our daily life because of the external genotoxic or any other stresses that cause the effects in cellular activities. Now, let me tell you what is the, let me talk about what is the P53. P53 is a transcription factor that acts as anti-tumor protein that checks the DNA mutations, different type of mutations in a normal cell and helps in maintaining the genome integrity. P53 control normally more than 160 genes. As it's a, it's a tumor suppressor gene. Now, what basically it does, when a human cell exposed to any type of the stress, either their UV radiations or whatever it is, this P53 get activates. The main tricky thing is that when this P53 activates, what it does, it suspends the cell cycle. The transmission of the cell cycle from G0 to G1 phase has been stopped by this P53. The basic activity of this is to suspend or to hold the cell cycle. The main reason for this, for holding the cell cycle, so that it can perform its own activity. P53 protein can activate three types of the main cellular responses: DNA repair mechanism, programmed cell death or apoptosis, or programmed cell death. But in this seminar, we are going to discuss about apoptosis and the programmed cell death and how it works or how it induced. Now let me tell you what is the basic structure of this. Now, P53 has a two mode of action: cell cycle arrest. What is the cell cycle arrest? When there are certain kind of mutations in the genomic sequences, P53 get activated. There is a gene product called ATM, attendant lactasia mutated. This ATM activates, triggers the response of P53. After the activation of the P53, it comes into the action. It having the 5-domain structure. P53 is approximately sequence of 393 amino acid. It comes into the action. It identifies the specific sequence where the mutation is there. After identifying that, there is a enzyme that cause the progression of the cell cycle. If you know the CDK1s, cysteine dependent kinases, that causes the progression of the G phase to the S phase for the progression of the cell cycle. What it does, when it identifies the mutation, it comes into the action and bind and releases certain factors that binds to the CDKs and suspend and activates the DNA repair mechanism. Basically, in normally 50% of the human cancer types, missense mutations has been identified. If these mutations can be correctable, then the P53 activates the DNA base excision repair mechanism that corrects the mutation, and after the correction, then the CDK become activated and the cell cycle progress to its normal state. If the mutations are irreversible, then it activates the apoptosis. After activating the apoptosis, what it does, basically there are two types of the main mechanism: intrinsic and extrinsic. We'll discuss this later on. Now, in this slide, it's been given that P53 induce the apoptosis that protect against the development of cancer and eliminates the tumorogenic cells. Means it eradicates the tumor cells and preventing our cells from mutations and from different kind of alterations to prevent our genome integrity. Now, this is in this slide you can see that this is a DNA damage. HDM2 is a transcription factor that binds with the P53 and phosphorylate it from three sites. It's 15, serine 15, serine 20, and 30. And after the activation, there is another protein called the P21 that comes into the action. Then further, this arrest the cell or the defected cell before progression from to the G2 phase. Then, now we are going to discuss about its domain arrangements. Domain basically P53 is a proper protein of about 393 amino acids having three regions: N-terminal region, central core region, and its C-terminal region. And each region having the total structure of this protein having the 5 domains. Now, the structure of P53 contains several trans activation, binding, and the core binding domain, tetramerization, and means each of this domain having its specific function. Now, we are going to check that what is exactly how does it work. Now, this is the structure of the P53. Here you can see it is the protein of length from 1 to 393 amino acids, having the three basic regions: N-terminus, central core, and the C-terminus. In the N-terminus region, there are two types of domains called the trans-activation domain and the proline-rich region. The trans-activation domains activates the activity of P53. Means when the mutations are occurred, when the missense mutations are occurred, then this trans-activation domain comes into action and activates it. Then the DNA binding domain containing the two specific consensus sequences that binds to the mutational DNA sequence and correct it. And the tetramerization domain is basically a domain that it binds four different P53 protein bind together through the tetramerization domain, forming a tetramer, forming a homoenzyme and regulatory enzyme. This regulatory domain enzyme, its basic activity